Size exclusion is a technique that separates proteins and other molecules by their hydrodynamic radius, by their not by their molar mass, but actually by their size. So within a column, which is packed with usually silica beads with several different pore sizes, things that are small get inside of the pores and start to get trapped in the column, so they elute later. And bigger things that can just pass through the little beads, they elute first. So it's a very nice way to separate proteins that are larger from proteins that are smaller, like aggregates from monomers. It's very important for quality control and for understanding how the protein behaves in solution. And together with size exclusion, what we do here, we associate the detector, which is usually a UV detector, with two other detectors that can measure the protein refractive index, which is the difference between how much the light interacts with the protein and how the light interacts with the solvent, and light scattering, which is a technique that measures when you shoot a laser beam through your protein, how that laser scatters the light. And that scatter is proportional to the molecular weight. This is our LC, our HPLC system, which delivers the sample to the column and to the first detector, the UV detector. And here on the side, we have the other two, which are the light scattering and the refractive index. So all these three elements together can give me a lot of information about proteins. The molecular weight, the hydrodynamic radius, and the diffusion coefficient in solution. This is the result from the size exclusion with the light scattering and the refractive index detectors that we were talking about. So in the green line here, you have the UV detection, which measures the protein aromatic residues like the tryptophan, the phenylalanine. In blue, we have the refractive index, which measures the protein concentration. It's directly proportional to the protein concentration. And in red, right here, we have the static light scattering. And in pink, this line here in pink, set of dots, we have the dynamic light scattering, which measures the protein diffusion coefficient in solution. So for this protein here, we have all the peaks overlapping, indicating that this everything is eluting together. And later on, I will make the data analysis to determine the protein molecular weight, extinction coefficient, hydrodynamic radius, everything that I can pull it off from this data. And that's what we do here in the quality control lab. We develop this type of methods that will improve our protein quality, our understanding about our protein, and the advance of our vaccine development.